In 1978, scientists were granted something that had been denied to researchers for centuries: permission. Permission to touch one of history's most sacred and controversial objects. Permission to take samples. Permission to study. Permission to apply the tools of modern molecular biology to an artifact that had spent nearly 700 years wrapped in mystery, legend, and faith. What they discovered in those samples would remain locked away in scientific archives for decades, analyzed but not fully understood, containing secrets that only the most advanced genetic technology could eventually reveal. That time has come. And what the DNA within those ancient threads has finally told us challenges everything we thought we understood about one of Christianity's most precious and contested relics. The Shroud of Turin is not simply a cloth, it is a mirror. It is the point where history collides with mystery, where science meets faith, where evidence confronts belief. For centuries, millions have looked upon it and seen a face, the imprint of a crucified man. And they have asked the same question over and over across generations and continents. Is this real? Is this the actual burial cloth of Jesus Christ? Is this the only remaining physical evidence of the central figure of Christianity? Or is it an elaborate medieval forgery, a work of human artistry and deception created to inspire faith and extract a devotion from believers? The shroud first appeared in the historical record in 1355, in the possession of a French knight named Geoffroy de Charny in the town of Lirey. Before that, its trail disappears into legend. According to tradition, it traveled through the ancient world, through Edessa in Turkey, through Constantinople, through Athens, through the medieval kingdoms of Europe. Each story grows more elaborate, more distant from historical documentation, more dependent on faith than fact. The shroud became a relic, a holy object, something that believers traveled thousands of kilometers to see, to touch, to stand before in reverence. Popes declared it authentic. Pilgrims wept before it. Artists painted its image. Theology was built upon the assumption of its truth. But in the modern era, science began to ask uncomfortable questions. If this cloth had truly wrapped the body of Jesus Christ nearly 2,000 years ago, it should tell a story. It should contain evidence. It should preserve traces of that ancient contact, protected by the very fibers that held the image. In 1978, a team of scientists conducted the Shroud of Turin research project, known as STURP. They examined every millimeter. They took photographs. They collected samples. They applied spectroscopy and chemistry and microscopy. And they found something remarkable. They found that every explanation seemed insufficient. The image itself defied conventional art techniques. No pigment could be detected, no paint, no dye. The image appeared to be created by something that oxidized the fibers themselves, browning them from within, as if the body had left its trace imprinted directly into the cloth at the molecular level. But the image was only the beginning. Locked within the fibers of that linen cloth were thousands of stories waiting to be told. Stories not of faith, but of chemistry. Not of theology, but of biology. Stories written in the universal language of DNA, the four-letter code that connects every living organism on Earth. For decades, those samples collected in 1978 remained in archives, studied but not fully unlocked. The technology simply was not advanced enough. The tools did not exist. But science does not stand still. Knowledge advances, capacity expands, and what seemed impossible 20 years ago becomes routine today. In 2026, a team of Italian researchers finally brought the full power of modern molecular genetics to bear on those samples. Gianni Barcaccia of the University of Padua and Alessandro Achilli of the University of Pavia led the investigation. They employed a technique called metagenomics, a sophisticated approach that allowed them to sequence every genetic fragment present in the dust and fibers, regardless of origin. Plant DNA, animal DNA, microbial DNA, human DNA, all of it, everything. They could not simply look for one specific source. They had to analyze the complete genetic tapestry, the accumulated biological signature of centuries of contact, handling, veneration, and storage. What they found was overwhelming. Not one genetic signal, but hundreds. Not a single source, but a chorus of voices speaking from the distant past. The data was so complex, so layered with contamination from so many sources that it told a story not of a single event, but of a journey. A geographical journey, a historical journey, a journey that the cloth itself had preserved in the code of life. The plant DNA told the first part of the story. Researchers identified 19 different plant species preserved on the shroud's fibers, pollen and plant matter clinging to the linen as it traveled through the world. Mediterranean plants, olive trees, fig trees, species native to the region around Jerusalem, species from Asia, species from the Middle East, species from the Americas, notably maize, which arrived in Europe only after Columbus, proving that the shroud's accumulated biological material came not from a single ancient time period, but from centuries of handling and exposure across multiple continents and eras. This contamination might seem like a problem to someone seeking to prove the shroud's ancient authenticity, but to a scientist, it was invaluable data. It was history written in the language of biology. It was a map, encoded in DNA of everywhere the shroud had traveled, everyone who had touched it, and when those touches had occurred. The shroud was not a sealed time capsule. It was a collector of evidence, absorbing the biological material of the world it moved through. But the most profound secrets were locked in the human DNA. When the researchers isolated and sequenced the mitochondrial DNA, the genetic code passed down from mother to child across thousands of generations, they began to trace the populations that had interacted with the shroud throughout its mysterious journey. Multiple human subjects had left their genetic fingerprints on the cloth. At least 14 different people of different ethnic origins, people from different parts of the world. The researchers identified several distinct mitochondrial DNA lineages. There was K1A1B1A, which matched the DNA of Pierluigi Baima Bellone, the researcher who had originally collected the samples in 1978. That was expected contamination from modern handling, but there were others, much older lineages that hinted at the shroud's ancient geography. H2A2, the lineage that serves as the reference sequence for all human mitochondrial DNA analysis. H1b, common in Western Eurasia and Europe. H33, a rare lineage, prevalent in the Near East and frequent among the Druze, a population with deep historical roots in the Levantine region, sharing genetic ancestry with ancient Levantine populations, including Jews, Palestinians, and Syrians. And then there was something that shocked researchers across the world. When the team analyzed the full spectrum of human genetic material preserved on the shroud, they discovered that nearly 40% of the human DNA traces back to populations of the Indian subcontinent, the ancient Indus Valley, the Indian lineages. This was not contamination from recent centuries. The genetic signatures suggested contact with populations from Asia, preserved on a cloth that, according to tradition, had traveled from Jerusalem to Constantinople to Europe. How did Indian DNA end up on the shroud? When did that contact occur? What does it tell us about the cloth's true path through history? The researchers themselves noted the profound implications of their findings. In their official analysis, they wrote, "A reevaluation of these DNA trace analyses on the Shroud of Turin suggests the potential broad exposure of the cloth in the Mediterranean region and the possibility that the yarn was produced in India. Not a proclamation, not a certainty, but a possibility, a hypothesis grounded in genetic evidence. The shroud may have been woven from Indian linen. It may have traveled through Indian populations. It may have absorbed the biological signatures of Asian peoples centuries before arriving in Europe." The microbiome told yet another layer of the story. Embedded in the fibers were microorganisms, bacteria, fungi, molds. Many of them were common human skin microbes, the microscopic ecosystems that cover every human body. But others were unusual. Archaeal communities adapted to high salinity environments, organisms commonly found in the Dead Sea, microbes that thrive in the harsh conditions of salt-saturated water. Why would such organisms be preserved on the shroud unless it had been stored in or transported through regions with such extreme environmental conditions? The bloodstains themselves told their own story. Researchers detected AB blood type, the rarest blood type, present in less than 4% of the world's population. The blood came from a human male. The genetic markers suggested ancestry connected to the Middle East, to the Levantine region. Was this the blood of the shroud's original subject? Or was it contamination from someone who had handled the cloth centuries later, leaving their biological mark? The science could not definitively say. What made this moment so extraordinary was not that it proved any particular theory about the shroud's authenticity or origins. What made it extraordinary was that, for the first time, science had managed to read the actual history of the object, not based on faith or tradition or legend, but based on the molecular biology preserved within its threads. The DNA was telling a story that no document could confirm, no archive could verify, but which was written indelibly in the code of life itself. When the Vatican received these findings, they issued a statement acknowledging the research. They noted that the shroud's passage through the Middle East was now confirmed by genetic evidence. They acknowledged that the cloth may have originated in India. They recognized that what scientists had discovered was not proof of faith, but evidence of history. A cloth does not lie. A molecule does not deceive. The DNA encoded in the fibers of the shroud spoke with a voice that transcended belief and disbelief, theology and skepticism. But the scientific community was quick to add important context. These findings did not prove that the shroud was authentic. They did not establish that it had truly wrapped the body of Jesus Christ. What they established was that the shroud was incredibly old, incredibly well preserved, and had traveled through multiple regions and cultures across centuries. The DNA showed contamination, from thousands of handlers. It showed biological material from many different time periods. It showed that the shroud had absorbed the biological signatures of entire civilizations. Some believers saw in these findings confirmation of their faith, the presence of Middle Eastern DNA, the possible Indian origins, hinting at the shroud's ancient journey following one possible tradition of Christ's relics, the rare AB blood type. To them, it was all evidence, all pieces of a puzzle pointing toward the truth they already believed. But skeptics noted something equally important. The contamination was so extensive, the mix of biological material so diverse, that any hope of isolating the actual DNA of the original body was essentially impossible. If the shroud truly contained the blood and tissue of its subject, that original biological material had been so thoroughly mixed and contaminated with genetic material, written over countless times, each layer adding new biological ink to the ancient parchment. What the DNA revealed, perhaps most powerfully, was not the identity or authenticity of the shroud. What it revealed was the story of how objects become sacred, how they accumulate meaning, how they gather to themselves the biological and spiritual testimony of millions of human hands across centuries of history. The shroud had been touched by crusaders and pilgrims, by priests and scientists, by believers and skeptics, by people from every corner of the medieval and ancient world. Each touch added their genetic signature. Each person who had held it, honored it, examined it, had left a microscopic trace of their existence on its fibers. The DNA analysis revealed that the shroud existed not as a sealed artifact, but as a living record of human history. Every population that had encountered it had contributed something. The Mediterranean peoples who had surrounded it, the Middle Eastern populations who may have possessed it in ancient times, the Indian weavers who may have created its very fabric. All of them, across centuries, had written their names into the genetic code embedded within the linen. Scientists emphasized that while the findings were fascinating, they changed nothing fundamental about the shroud's ultimate question. The great mystery remained. Does it bear the imprint of Jesus Christ? Is it one of history's greatest relics or one of history's greatest artistic forgeries? The DNA could not answer that question. It could trace geographical origins. It could identify plant species and bacterial communities. It could place the cloth in a historical context, but it could not distinguish between an authentic burial cloth and a medieval production that had been passed through the same regions and handled by the same peoples. Yet, in a sense, that limitation was itself profound. It revealed the boundary between what science can know and what faith must believe. The shroud exists at that boundary. The image itself defies conventional explanation. The preservation is extraordinary. The journey through history is remarkable. But whether all of this points to the sacred or to the ingeniously artistic remains a question that no genetic sequence can definitively answer. Themselves the traces of every life that touches them. It is a reminder that history is not written only in documents and monuments, but in the microscopic code of life itself. The DNA evidence does not solve the mystery of the shroud, but it transforms the mystery. It moves it from pure speculation into the realm of verifiable biological fact. We now know that the shroud contains genetic material from the Middle East. We know it likely originated in or passed through India. We know that Mediterranean peoples held it. We know that countless individuals across centuries left their mark upon it. We know the shroud's biological story, even if we do not yet know the full truth of its spiritual significance. Standing before the shroud of Turin today, a visitor sees not just an image, they see something that has become a repository of human history, a cloth that has been held by saints and sinners, by believers and skeptics, by people from every walk of life and every corner of the earth. The DNA within its fibers whispers their stories. It speaks of journeys across continents. It speaks of centuries of handling and veneration. It speaks to the profound human need to touch the sacred, to reach across time and connect with something greater than ourselves. Whether that cloth is authentic or not, one thing is certain. It matters. It has mattered to millions. It continues to matter. And now, through the lens of genetic science, we can see not just what the shroud claims to be, but what it actually is. A time capsule, a biological archive, a testament to human faith and human history, written in the universal language of life itself. The shroud's greatest mystery may no longer be what it is, but what we are willing to believe about what science reveals to us.